Hey there, we're going to talk about uh, genetics and the discovery of DNA. So in this first video, we're going to talk about the first part of chapter 16, the discovery of the DNA molecule. And so we have to understand and remember that any discovery in science is compounding, and that while we credit Watson and Crick for the discovery of genetic material, there were a lot of previous and elegant labs and discoveries made before they came to their conclusions. And so we're in the time after T.H. Morgan and Sturtevant, if you remember from our previous chapter, how T.H. Morgan and Sturtevant came to the conclusion about gene linkage. Um, we have determined that there are genes on chromosomes and they're linked and they're not linked, but we don't know what these chromosomes are. We don't know what this molecule is. And so there's two hypotheses, but most scientists believe that it's proteins. Proteins are a very diverse molecule with very specific functions. There's so many of them and there's just so many different traits. So it made sense that proteins, with their very diverse nature, would be the genetic material. However, nucleic acids were in the game. However, they seem too uniform, just A's and T's and C's and G's, and they're just bland. And how could they provide all of the diversity of life? It had to be proteins. And so it was a race to figure out the which molecule it was. And so our story starts with Frederick Griffith in 1928. He was working to create a vaccine against pneumonia. And so he did this elegant experiment here where he was working with mice and with bacteria. And so there was pathogenic bacteria, and he injected pathogenic bacteria into a mouse, and that mouse died. That was his control, and that makes a lot of sense. Pathogenic bacteria into a mouse, the mouse dies. And his other control, non-pathogenic bacteria, he put that into a mouse and the mouse was healthy. That makes sense too. So these are great comparisons for his control. So what he did next was he took his original pathogenic bacteria and he killed it. So it was dead, and then he injected it into a mouse, and that mouse survived. And so still this makes sense. So he killed the pathogenic bacteria, put it in the mouse lived. So it gets surprising over here when there was a mixture of, he took a mixture of both the pathogenic, heat-killed pathogenic bacteria, and then the non-pathogenic, living non-pathogenic bacteria. So he mixed those together and put those into the mouse. And what he found, and he was surprised, was that the mouse died because it didn't make sense because the pathogenic bacteria was heat killed. So how is it hurting the mouse? What he found and discovered was that there was a transforming molecule, that somehow there was a transformation that was going on between the non-pathogenic or from the pathogenic to the non-pathogenic bacteria. And so this transformation, it changed the genotype and phenotype of the non-pathogenic bacteria to make it a pathogenic bacteria. And so he didn't know what the molecule was, but he says that there was a transformation. There was some sort of transforming molecule. But what was it? Later on in the 1950s, Hershey and Chase come up with another elegant discovery, and they were using bacteriophage. So bacteriophage are literally bacteria eaters. Phage, eaters, bacteria, they're eating bacteria. And what they did was they used the genetic material of the phages, injected it into bacteria, and determined which molecule it was. So let's go to this slide here. So here is our phage, and we put it on a bacteria. So here's our bacteria, and what it does is it's going to inject, this bacteriophage is going to inject its transforming molecule into the bacteria. And the bacteria, what it does then is it uses those instructions to make more and more and more bacteriophages, so many that it explodes, just spewing out bacteriophages all over the place. So pretty impressive form of reproduction on the bacteriophage's part. And so bacteriophages are composed of not many materials: a protein coat, which we can see here, and then genetic material on the inside. We know this is genetic material now, but we didn't know that it was a genetic material back then. And so we did know that proteins have sulfur, and DNA has phosphorus. While proteins have sulfur, they don't have phosphorus. And while DNA has phosphorus, they don't have sulfur. So Hershey and Chase radioactively labeled the virus with radioactive phosphorus in the DNA and radioactive sulfur in the protein. And they let the virus or the viruses infect bacteria. And so they were injecting the bacteria with the transforming molecule. And so they needed to figure out, what is that transforming molecule? It's now in the bacteria and no longer in the phage. So is it going to be sulfur or is it going to be phosphorus? If it's sulfur, it means that the transforming molecule is a protein. If it's phosphorus, it means that the transforming molecule is DNA. So they used a blender to agitate and to separate the viruses from the bacteria because they're stuck on them and they needed to separate the two. But then you have a mixture of both phages and bacteria. So they use a centrifuge, spin it around, and it takes, so a centrifuge takes test tubes and spins them around to separate different parts of a solution. And so in this solution, the supernatant, which is the liquid on the top, was filled with phages because they're less dense. While bacteria is more dense, it was in the pellet in the bottom of the test tube. And so the bacteria that's in the pellet had radioactive phosphorus in it. So this part with the transforming molecule is down here in the pellet. And so that told us that the radioactive phosphorus is in fact in the DNA, and DNA is a transforming molecule. Then they found that the radioactive sulfur was in fact in the supernatant. And so that undoubtedly DNA is our transforming molecule. So it continues on. In 1950, Chargaff knew that DNA had three components: phosphate groups, ribose sugars, and nitrogenous bases. However, he continued to analyze the composition of the nitrogenous bases, and he found that a couple of things. One, that there's a molecular diversity among different organisms. So he found that a sea urchin will have about 34% adenine, while humans have 24.7% adenine in their DNA. And so that means that there's variation in the genetic material between organisms and between species. And then he also found that there was similarities in the amounts of adenine and thymine, and that there were similarities in the amounts of guanine and cytosine. He didn't draw any conclusions on this. He just made the statement that there are similarities in these materials, in these numbers of materials. But he did not conclude the base pair rule. In walks Rosalind Franklin. She used X-ray crystallography to create this picture, and this picture shows us the basic makeup of DNA. It shows that it has that it turns. And so here you can see that DNA turns. If you were to hold your right hand up like this, that looks like right to you. If you hold your hand up, you can see here that that's the direction that DNA is going to turn. And that one turn is 3.4 nanometers in length. And it's made up of 10 base pairs. And so it makes sense that each of the base pairs are 0.34 nanometers. And so this was quite a bit of information about the structure of DNA. And so with that information, Watson and Crick used the discoveries of Rosalind Franklin and also of Chargaff. They found that the sides of the molecule are anti-parallel. One thing that Rosalind Franklin also discovered is that there were 2 strands in DNA. Not 3. There's a hypothesis at the time that DNA was a 3-stranded molecule. Rosalind Franklin, with her picture, discovered that it was a 2-stranded molecule. Watson and Crick discovered that they were anti-parallel, that those 2 sides were anti-parallel. They used Chargaff's information about A's and T's and G's and C's having similar ratios, and understanding that from Rosalind Franklin's picture that the 2 sides were uniformly distant. So putting everything together like a puzzle, like you can see here, they discovered that there were purines and pyrimidines, that there were bigger molecules and that there were smaller molecules. I remember pyrimidines is a longer word and it's a smaller molecule. It has 1 ring, whereas purines is a shorter word and a bigger molecule. And so a bigger molecule is always going to pair up with a smaller molecule. So a purine will always pair up with a pyrimidine. That way you can maintain the even shape or the even distance along the whole molecule, because if you had 2 pyrimidines, it would be smaller in some sections. If you had 2 purines, it'd be larger in some sections. And so therefore Watson and Crick came up with the complementary base pair rule, that A's and T's are going to pair up and G's and C's are going to pair up. Okay, so that is 16.1, and I will see you for 16.2.